Hello and welcome for a webinar on genetics and heredity. We have studied yesterday the basic principles, basic concepts of genetics, and today we will proceed further with the study of genetics and heredity. Hope you all remember what is genetics, who is the father of genetics, and so on. Father Gregor Mendel, he was a Christian priest, so he can call him as a Father Gregor Mendel, and he discovered modern genetics. And today we are going to discuss this genetics and heredity in a further depths of the topic. Okay, so I welcome you back for this today's session. Let us understand what are the different genes, what you call dominant gene and recessive gene. Dominant gene is more powerful, whereas recessive gene is weaker and hides in the background. That I have told you yesterday also. Now also we spoke about that. And there are two other pair of genes, carrier and sex link. Carrier gene has a recessive that is not visible in that person. And sex link means mother passes the recessive X to son. For example, the mother is usually not color blind herself because she is carrier, but she will pass it to her son. The color blind male receives the trait from his mother. This I have explained yesterday also, and today also we are taking it for the sake of background study. And what is heredity? I told you heredity means traits, characteristics which are seen in the offspring. For example, if we give example, common example is of eyes, brown eyes and blue eyes. The uppercase B stands for dominant gene, that is brown, and lowercase b stands for a recessive gene, that is blue. If there is a combination of these T, double B capital will give you always brown, small b will give you always blue. But if there is upper B and lower B, or big B and small b, then big B will play a role in giving color to the eyes, and that is always brown. So don't forget this example. It's a very common example. Even though the big B brown is the color eye, but still carry the recessive blue gene with it. Okay, and human genome is here in front of you. Everybody know this, that human cells contain 46 chromosomes, 2 X chromosome, one pair of chromosomes, sex chromosome, and 22 pairs of autosomes. That everybody know. In case of males, it is 44XY. In case of females, it is 44XX. I am repeating this because it's very important. Finally, you have to remember genome. Genotypes and phenotypes, small definitions are here in front of you. At each locus, there are two genes, and these constitute the individual's genotype at the locus. And whereas phenotype, the expression of a genotype is termed as phenotype. For example, color of hair, the person's weight, or the presence or absence of any, heredity diseases. That is what is called as a phenotype. And because we are also going to study today some genetic disorders, chromosomal disorders and genetic disorders, that's why I'm giving you these examples. Now, genotypes and phenotypes examples, if you see here, is one example. In upper row there are genes or alleles shown here, and in the lower row there are pictures of flies with black eyes and white eyes and black eyes. So this is a phenotype and this is a genotype. Another example is example of dominant versus recessive gene. This is example of cat. There are brown cats and there is a gray cat here. Brown stands for B uppercase for brown color and B lowercase for gray color. Both the B will always give you gray color, and both the uppercase B will give you always brown color. But if the combination is uppercase B and lowercase b, here the uppercase B color will play a role. So this cat is having brown color. It's a very small example. A dominant allele is expressed even if it is paired with a recessive allele, whereas recessive allele is only visible when it is paired with another recessive allele. So for example, the small case BB. So these are the very small, common examples of genetics. When the uppercase is paired with uppercase is paired with lowercase a, it is called as heterozygote. Heterozygote. When the lowercase a is paired with lowercase a, it's called as homozygote. It means same to same. And dear students, there are two common laws of Mendel's, because it is genetics is postulated by Gregor Mendel. The Mendel's first law states that two members of a gene pair segregate from each other into the gametes. So half the gametes carry one member of the pair and the other half carry the other member of the pair. So very simple, first Mendel's law. And calculating the probabilities, we have seen lots of examples yesterday how to calculate the probabilities. I have given you lots of examples yesterday. We want to predict patterns of inheritance of traits and diseases in pedigrees. For example, we want to the likelihood that a dog chosen at a random from the study population will have blue eyes. So this is example of probabilities regarding color of the eye. So let us proceed further. Now X-linked inheritance. This is more related to X-linked diseases. Different results obtained from reciprocal crosses between red-eyed and white-eyed Drosophila. Experiencing the gene response for eye color is X-linked. Females have 2 X chromosomes while males have 1 X chromosome and 1 Y chromosome. So common example I have given you here regarding X-linked inheritance. Let us proceed further regarding Mendel's second law. Different gene pairs assort independently in gamete formation. Okay, this law is only true in some cases. It's an uncommon law. Gene pairs on separate chromosomes assort independently at meiosis. Next is genetic defect. Now today's, I'm giving more emphasis on genetic defects because we are more interested in medical genetics. Okay, the genetic defects are either X-linked or sex-linked or sex-linked or X-linked, either way. When an X gene from the mother is faulty, there is a 50-50 chance of the child inheriting the disorder. The best example I have given you earlier. Now next one is syndrome. Syndrome means signs and group of signs and symptoms seen due to some genetic disorders or genetic defects or chromosomal disorders. When a group of signs and symptoms occur together and characterize a particular problem, so that is called as syndrome. For example, Down syndrome, Klinefelter syndrome, Jacob syndrome, so many of them are there. Congenital malformation, a condition that is present since the birth. For example, if person is having a 6th digit sour boat, or if there is a cleft palate or cleft lip, something like that. So that is called as congenital malformation. And multifactorial defects, that is interaction of genes with other genes or with environmental factors. And last is chromosomal defects. So genetic defects and chromosomal defects, there are two different categories. The fertilized ovum that contains chromosomes in an abnormal number. For example, here abnormal number is all the Down syndrome, Klinefelter syndrome, and so on. Okay, and in X-linked we have got that hemophilia, what you call cystic fibrosis and so on. That we will study now one by one. Now let us deal with medical genetics, which is a closely related topic to us. This is about diseases and disorders and diseases. More you have to study those in principles of practice of medicine. But for the first year students, because it is related to genetics, we have to remember these all names. When studying rare genetic disorders, 6 general patterns of inheritance are commonly observed. One is autosomal recessive, second is autosomal dominant, third is X-linked recessive, then fourth is X-linked dominant, then codominant and mitochondrial. So these 6 categories or 6 patterns what we see. Now one by one, let us proceed. First is autosomal recessive. The autosomal recessive, the disorder main is cystic fibrosis. Example, please see the example. In this disease, the disease appears in male and female children of unaffected parents. So both the parents are unaffected and carrier father and carrier mother, they are carrier. Then in case of that, what probabilities you get, you can see at the end of bottom of this image. Unaffected son, carrier daughter, carrier son, and affected daughter. So this is very what you call problematic here, because this girl is going to suffer from cystic fibrosis, and whereas this girl is going to carry, this boy is going to carry, and he is normal here, unaffected son. Okay, so this is about autosomal recessive defect. Cystic fibrosis. Now, what is cystic fibrosis? What we saw in this example, there is recessive. This is recessive disorder. The mutation stops production of protein in the lungs, and thick mucus bacterial infection in lung take place. And sweat test is for screening. Most common in Caucasians. Caucasian means is a race in Europe. 1 in 33, sorry, the 100. Chest percussions, diet supplements is the treatment. Shorter life expectancy, so they usually die early. So this is about cystic fibrosis, and this is a genetic defect. Now next is autosomal dominant. In autosomal dominant, what we have commonly, example is Huntington disease. So this Huntington disease, similar kind of image is given here to study affected father, unaffected mother, and the combination you can see. And here you can see affected son here suffering from Huntington disease. In this disease, affected males and females appear in each generation of the pedigree. Affected mothers and fathers transit the phenotype to both the sons and daughters. So that is what is explained in this image. Now we'll go to the next, the details of Huntington's disease. What happens in Huntington's disease? It is also called as SD. It is a progressive brain disorder caused by a defective gene. This disease causes changes in the central area of the brain, which affect movement, mood, and thinking skills. So this is Huntington's disease. Now we'll go to another, disorder, X-linked recessive. In X-linked recessive, the common example is hemophilia, and hemophilia patients we get a lot. Here in this case, X-linked recessive gene. Usually the unaffected mother is a carrier. I told you in the beginning also, and father is unaffected. And here you can see carrier daughter, unaffected son, X-linked recessive carrier mother. If the mother is carrier, then the son is affected. This example we have spoken earlier. What happens in this case? Many more males are affected than females show this disorder. All the daughter of the unaffected male are carriers, and none of the sons of affected males show the disorder or are carrier. For example, hemophilia. Now what is hemophilia? You can see one image here. It's a bleeding disorder. It is sex-linked, X recessive, failure of blood to clot. So there is no blood clotting possible in this case. It is rare in females. So it is called as, it is common in males. Now injections with clotting factors to stop bleeding episode is the only remedy for this. And the treatment is very expensive because again and again, all that blood supplement has to be given to these patients. So there are hemophilia clubs there to keep such patients alive, because always they keep on bleeding. Okay, and X-linked dominant. Now next example is Fragile X syndrome. In this case, affected males pass the disorder to all daughters but to none of their sons. Affected heterozygous females married to unaffected males pass the condition to half their sons and daughters. Here you can see the images shown here. X-linked dominant affected father, and below is X-linked dominant affected mother. And this is the outcome, the lower line. Preferably, because of time constraint, I cannot explain each and every slide in detail, and for first year level, for first year, everything is not that much essential. You try to take screenshots on your mobile, Android mobile. That is very easy for you, and you can later on study with the reference books. Now Fragile X syndrome means what? Most common inherited cause of mental retardation. So these offspring, these children, they suffer from mental retardation. Okay, and it is 1 in 2,000 males. It occurs 1 in 2,000 males. High forehead. If you look at this boy, he is having high forehead, prominent jaw. You can see this jaw is prominent, and autism. They have got mental retardation, gap in X chromosome. It is characteristic of this. Now we move on to next category, that is codominant inheritance. Now ABO locus is the example of this disorder. Two different versions allele of a gene can be expressed, and each version makes a slightly different protein. Both alleles influence the genetic trait or determine the characteristics of the genetic condition, and codominant example is shown here. Okay, and ABO locus is the disorder. Now we will go and see what is abiolocus. Now abiolocus means in which the catalyzes the transfer of gall residues from UDP gall donor nucleotide to gall residues of the acceptor H antigen, converting the H antigen into B antigen and B and AB individuals. So in this case, you can see it has got 6 possible genotypes, and they lead to 4 possible phenotypes. So problem phenotypes doesn't reveal genotype. This is about abiolocus, very rare. Now we will go to next category, the last in this in this study, that is mitochondrial inheritance, and the disease is LHO1, what you call Leber's hereditary optic neuropathy. The eyes are involved in this case. This type of inheritance applies to genes in mitochondrial DNA. Okay, and mitochondrial disorders can appear in every generation of a family and can affect both males and females. But fathers do not pass mitochondrial traits to their children. Here you can see the example, mitochondrial inheritance. Unaffected father, affected mother, affected father, unaffected mother. And here is the outcome. Affected children, all of them. Unaffected children, all of them. Okay. Now we will move to see what is LHON. Okay, from mitochondrial inheritance. So it is often characterized by bilateral painless subacute loss of central vision. So these people can't see during young adult life. So in most cases, symptoms begin with one eye first and followed a few weeks later by visual failure in other eye. So one eye gets affected first, then other eye. So it is in the adult life they lose vision. So this is the characteristic of this disease, that is LHON. Okay. Now we have finished that topic. We'll go to single gene disorders. And in single disorders, we have 4 examples: cystic fibrosis, hemophilia, sickle cell anemia, and phenylketonuria. Upper to cystic fibrosis and hemophilia we have discussed in above category. So we will not discuss that again now. We'll just talk about sickle cell anemia and phenylketonuria as far as the single gene disorder is concerned. Now, sickle cell anemia is due to mutation of gene in blood protein. Okay, when the RBC becomes sickle in shape, you can see this is a what you call biconcave. No, biconcave RBC is a normal shape, and here you can see the sickle cell. This is affected cell. The screening test uh, is done, most common in African American people. Pain is associated with blocked vessels. Due to sickle cell, the blood vessel gets blocked and it causes pain to the patient. It also causes anemia. It causes fatigue because there is a hypoxia, low oxygen level. Common where mosquito borne malaria is present. So it is more common there. And that is why sickle cell anemia disease is, genetic disease, because there is mutation of genes. That is why we have taken this in, we are considering this in this study. And now last one in this category is phenylketonuria. Phenylketonuria PQ we have talked about in the study. Mutation disrupts functions of enzyme, leads to high phenylamine levels in brain, that is poisons, and there is mental retardation and epilepsy. The screening test is done in newborns by heel prick. You can see this picture here. Heel prick is given, and extremely rare in African Americans, but it is common in Caucasians. The child looks normal, need low protein diet and smelly formulas. This is about phenylketonuria. You just try to remember at least the names. And at the end of this study, there is a small quiz. You should be able to answer what you call MCQs. Now we will go to the last part, that is autosomal disorders. Autosomal disorders here, I'm giving you 3 examples: Down syndrome, which is trisomy 21, Pato syndrome, trisomy 13, and Edwards syndrome with trisomy 18. At the level of 21st, chromosome, there is a problem. At the level of 13, there is a problem. At the level of 18, there is a problem. These are 3 different things. But the Down syndrome is very, very common. In Down syndrome, what you call DS, there is excess 21 chromosome. So there is one autosome extra here in this case. Prenatal testing can be done before birth by amniocentesis. I'll show you that case later on. And result of chromosomal mutation causes Down syndrome. 1 in 900 people born with this. So this is very common. See, 1 in 900. likelihood of having a child with Down syndrome increases with advancing maternal age. So those women who give birth to the children at the advanced stage, they are having more chances of getting Down syndrome babies. Symptoms in Down syndrome, what we see is mental retardation, upward slant eyes, small mouth, abnormal ear shape, decreased muscle tone. We have discussed it yesterday also, and there is no cure for this condition. Okay, they remain as they take birth till they die, and the life expectancy is also very low. They are infertile. Now, Patos syndrome or Edwards syndrome, together we take both of them. In this case, there is a cardiac abnormalities. They are very severe conditions. Most affected infants die. Soon after birth, they die during the first few weeks of life. So there is no chances of survival of these two syndromes. Now we have finished that topic. I will go to deletion disorders. One is Angelman syndrome and that is Prader-Willi syndrome. What happens in Angelman syndrome is inappropriate laughter with convulsions. So this is, you can see this small girl here. She is suffering from Angelman syndrome, poor coordination and mental retardation. This is due to deletion disorder. So there is a deletion of segment of chromosome in that one, and other is Prader-Willi syndrome. Here, these children are usually extremely floppy. Floppy means obese. Obese means fat. Constantly they are hungry. They keep on eating and putting on more weight. There is a mild mental retardation also in such case of patients. And the last one is sex chromosome disorders. And this is very easy. You have seen yesterday. I have shown you this yesterday also. Klinefelter syndrome, Turner syndrome, Fragile X syndrome, and Jacob syndrome. So we will see one by one. In case of Klinefelter syndrome, there is one X chromosome extra in sex chromosomes, or making it all together 47. So 1 in 1000 male live births, there is a mild learning difficulties. They are taller than average with long lower limbs. The lower limbs are very long. Show mild enlargement of breast. Even though boys, they have enlargement of breast, and they are infertile because absence of sperm. These are to be treated with the testosterone injections. So Klinefelter syndrome is one extra sex chromosome here. That is X. So they look feminine. I told you yesterday about it. Now Turner syndrome is, here there is one sex chromosome less. So it is 44 plus X. One X is missing. It is only present in the girls. So one X is missing. And this is a low incidence. Right? It happens less often. They look normal, but there is ovarian failure and no fertility, normal intelligence, but they have a short stature, unchi kami aste, webbed neck. So soon after head there comes a thorax, and estrogen therapy treatment can be given due to exchange of 1X. Next is Fragile X syndrome. I have talked about this earlier in upper category. Most common inherited cause of mental retardation. High forehead, prominent jaw, autism, that I have told you earlier, so not to waste time. And the last one is Jacob's syndrome. Jacob's syndrome is just like Klinefelter syndrome, but here what happens? There is one Y more. In Klinefelter syndrome, there is one X more. In Jacob's syndrome, there is one Y more. So that means Klinefelters are more feminine, and Jacob's syndrome more masculine. That is why this happens only in boys, and the boys become very tall. You can see this example, and here you can see this one X, Y, Y. Okay, and males normally have one X chromosome. However, individual syndrome have one X chromosome and two Y chromosome. Affected individuals are usually tall. Multiple births means more than one child, like twins and triplet and quadruplet, and conjoined babies means Siamese babies. These are not genetic or chromosomal defect, but these are multiple births. We will deal about it when we go for study of embryology. And these are the other factors which are responsible for multiple birth. So it is not a genetic or chromosomal. Okay, it is due to high age or some drugs or history in the family. And next one is these are the tests: chorionic villi sampling and ultrasound to detect all the genetic and chromosomal disorders. Amniocentesis is done. And now first MCQ, you can see which of the following X-linked disorder is hemophilia. In PQ there is, you have to go for heel prick. In CF you have to go for chest percussions. In Turner syndrome, it is 45x. In patients with Klinefelter, it is all males. Some of the examples I am giving you fast due to time constraint. Medical termination is done in 6 months. Sickle cell anemia is the mutation of genes. Drugs used for sickle cell anemia is hydroxyurea. Blackening of urine is in PKU. Crisis in sickle cell, all of the above. So these are the some MCQs which are there. I'll take those again because of time constraint. I'm going very fast because the light gave us what you call a little trouble today. And with this I end my today's topic and see